So looking at individual levels, at individual level, a human being is made up of a huge amount of cells, about 30 trillion cells in an individual. I'm not going to bug you with the number, but what I want you to remember is that you have a large number of cells, and these cells, they are not just numerous in terms of number, they are also very diverse in terms of the kind of cells there are, right? You're going to have muscle cells, you have skin cells, you have liver cells, you have eye cells, okay? You have nerve cells, you have liver cells, you have cells of the intestine, and so on and so forth. You have a wide diversity of cells. In the cells, though, you have some cells that are specialized. These cells that are specialized are what we call differentiated cells, okay? Differentiated cells. And you have cells that are non-differentiated cells. The cells that are non-differentiated cells are going to be what we call stem cells. These cells can divide over and over and over and produce themselves again and again and again, right? These stem cells is where individuals start, right? You have a sperm cell that meets an egg cell. You have the formation of a zygote. That zygote is going to divide into stem cells, and the stem cells are responsible for the wide diversity of all the cells that make up you, okay? Now, eventually, because of the differentiation of stem cells, you have that cells are now going to specialize in doing something very specific as a function and occupying a very specific space, right? For instance, a skin cell can only be found in the skin. It cannot be found in the muscle tissue. No, that is not going to happen, right? Then the cells are going to come together based on their structure and function to form tissues, okay? Similar cells working together toward a specific function. And tissues are going to come together to form organs. Organs are going to come together to form organ systems. And all the organ system in your body make you, right? Now, looking a little more into genetics in terms of genetic makeup and the outcome of gene expression, we are going to use the term genotype. A genotype is basically what you are made up of as genes, right? The different alleles. Which allele did you receive from mom? Which allele did you receive from dad? Right? Together, that's your genetic makeup. Eventually, that genetic makeup is going to express. And when that expresses and produces an outcome that is quantifiable, that is observable, now we are talking about a phenotype. Okay? Without expression, you don't have a phenotype. Okay? Now, the alleles that make up an individual can be dominant or they can be recessive. When you have these alleles, a dominant is going to be an allele that gets expressed. A recessive allele is an allele that doesn't get to be expressed, right? Now, considering individuals, we can now try to look at, how is a particular individual related to what we think, who we think are their parents and their grandparents and their great-grandparents. How are they related, right? Now, not just looking at the social aspects, but looking at the genetic aspect, right? Now we can talk about what we call a pedigree, right? A pedigree is a chart that basically describes how family members are related. Now, when it comes to genetic genealogy, we are going to really see, okay, well, how are the traits going from great grandparents to great grandchildren, right? So we have, say you are right here. You are made up of 50% of your mom and 50% of your dad. See here, you have your mom, okay? Here you have your dad, okay? So then you represent 25% of each of your grandparents, okay? Now you have grandparents, your mom's side, your dad's side, right? And you can actually look at your genetic affiliation up to your great grandparents, where you are only related 6.25% of each of your great grandparents, right? This information becomes very relevant when it comes to, for instance, you want to prove that, oh, I have Native American background, right? So what percentage? All right. So taking a look at the big picture here, we are going now from individual level to population level to evolution, right? At the population level, we are going to define a population in biology as individuals that were developed, right, by groups of individuals that were able to reproduce among themselves and leave behind offspring and forming a group of people, right? That's the very basic biological definition of a population. But when it comes to genetics, population is not defined as the number of individuals that were born from X, Y, or Z and living together. But we are going to take a look at genetic level. We're going to ask the following. When we consider this group of individuals, what is the presence of a particular allele? What is the frequency of that allele, right? Allele A, capital A, or allele lowercase a, what is the frequency? So that when we consider a population, we're going to consider a population as a collection of genes, a collection of alleles, okay? What we call a gene pool. A population at the genetic level is defined as gene pool. That's very important to understand. And from that gene pool, we are going to ask the following question. Has anything changed in the gene pool when we look at the population today compared to the population 50 years back? Has the gene pool changed? If the gene pool has changed, then we are going to say, there is actually what we call evolution. Okay? There is evolution. Now, that evolution, it's basically depicted by the change in the frequency of allele in the population. Now, when you consider a country such as the United States, you have a lot of immigration and emigration in the United States, right? And out of the United States. So because people are coming in the United States and people are leaving the United States, well, you are necessarily going to have evolution in the population in the United States. But let me close that parenthesis. We're going to develop some more about that later. Let's take a look at application of genetics and genomics. Well, application of genomics and genetics, I already mentioned some of the aspect, but to be specific, genetics can impact your life in ways that I'm pretty sure you understand how deeply it can impact your life. In terms of your healthcare choices, in terms of what you eat, in terms of your past, and also your future. How so? For instance, getting to know your genetic information is going to tell you if you are prone to cervical cancer or breast cancer or prostate cancer, which is going to necessarily, if you do care about your health, you're going to be like, okay, well, you know what? There are just some jobs I'm not going to ever take because they put me at a risk for developing a certain disease, right? There are some things that I'm not just going to consume, like alcohol. I'm not going to drink alcohol because it is known that alcohol is prone to trigger some diseases in some conditions, right? So, knowing your genetic background is going to affect your life in a way that you wouldn't anticipate, right? Today, we talk more and more about citizen scientists. Citizen scientists, it's basically everyday people taking responsibility, right? Going about gathering information about themselves, turning themselves into scientists, right? Trying to understand, right, what they are at risk of developing or not. And that has become very important, not just for the individual, but also for the community as a whole, right? So that we can anticipate and we can tackle some issues. Now, my question to you is this. When you hear genetics, what comes to your mind? This is a question I want you to think about. Do you have the expression of fear? Is it a certain hope that maybe we can solve some problems that we didn't even know they existed until some problem happens to us, right? Is it anger? Do you wonder what is going to happen with the information that we get? Or is it like, oh man, this is too bad? Despair? Okay? How is DNA information used today? I want you to think about that. And also, I want to discuss a little bit, just a little bit, about the COVID vaccine, okay? The Pfizer version. Now, when the COVID vaccine was developed, a lot of people thought, okay, well, wait a minute. Why was the COVID vaccine developed so fast? Why was it developed so fast? Is the government trying to get us somewhere? Or are they trying to control me? What are they trying to do? Now, here is one information that many of you may not know. While people were living their lives before COVID-19, researchers have always worked on infectious diseases, including viral infections. It so happened that people were working on the coronavirus before the pandemic, but who cared about it, right? The average person didn't care about it. Politicians didn't care about it. Most people didn't care about it, except that scientists are sometimes very stubborn, right? They know that, okay, this is not even allowing me to live a decent life, but you know what? I'm driven by what I'm interested in. I want to try to understand this. Some scientists actually spent a lot of time studying the coronavirus and actually developed ways to fight the coronavirus. But who knew about that? Certainly not somebody who is not a scientist, right? But me as a microbiologist, I knew that people worked on coronavirus. And when the pandemic occurred, it was no surprise that all the information people had gathered for many, many years, it was just a matter of putting that information to application. And people didn't understand that, well, scientists already had a great deal of knowledge. Same thing today. When you go to the hospital and you get screened for cervical cancer, and you are told that, oh, well, there is a chance that you may develop cervical cancer. Well, today you are not wondering anymore, wait, how can you be so sure about that? Well, you are not wondering anymore because we talked so much about it, right? About screening for cancer. But who knew about screening for cancer 50 years back? Well, maybe not 50 years back, 100 years back, right? We didn't know very much about that. But today, in a matter of hours, you can be told that you are at risk of developing cervical cancer, you are at risk of developing prostate cancer, and so on and so forth, right? Think about that. Now, it is possible to establish a clear identity of in the relationship you have with your surrounding, the people around you. By just doing comparative DNA analysis, we can tell that, I didn't know that I was related to you. Okay? Now, we know that in terms of history, the history has told us major things that we thought were there, but we didn't have proof. I will come back to that. With DNA analysis, we can now apply that to forensic science in terms of identifying who was the criminal in a particular case. But people who have claimed their innocence for many years and who have put in jail, we can also show that, hey, they were not the criminal, right? You need to free them, right? Okay? We can identify victims in accidents or in natural disasters. We can actually determine if what we call beef, it's beef and not a horse. Because believe it or not, in the craziness, in the race of who is going to become rich, right? People, unfortunately, have engaged in criminal activities, including selling something for what they are not, right? Saying that this meat is beef when that meat is not beef, it's actually horse. Think about that, right? These are real stories that are happening even in the United States, right? So, especially in the world of processing of food, right? So we know today that, for instance, if you consider somebody like Thomas Jefferson, that for a long time, the nation thought, well, we think that Thomas Jefferson had children with a slave. And some people argued, no, that didn't happen. That is not possible, right? But the fact is that with DNA, it was possible to show that people who actually were clearly not Caucasian carried Thomas Jefferson DNA for real. And now you have this photograph that shows you a family reunion that happens yearly for the Jeffersons, right? And you can see a wide diversity of individuals here who actually can claim their relationship to Thomas Jefferson, one of the founder person in this country. Because of genetics, now we can talk about precision medicine. Now we can say, well, it is true that you have this particular disease. It is true that we have this particular drug to treat that disease. But guess what? We're not going to use that drug with you because we want to know specifically what works best for you rather than giving you a generic medication that we won't even know if it's going to work on you or not, right? We call this pharmacogenetics. So pharmacogenetics allows us today to design treatments that are very specific to work on a particular patient and effectively on a patient. We talk today about microbiome, which is actually all the characterization of all the microorganisms that are in you and on you. We know that some diseases, like dementia, that we didn't suspect that they would be associated with your microbiome are indeed associated with your microbiome. Okay? But let me close that parenthesis. Now, because of all the knowledge that we have in genetics, it is possible to modify the genomes using different approaches. That genetic modification allows us to improve living organisms, allows us to produce enough food for everyone, allows us to produce medications. Think about all the cancer drugs that are produced. Many of them are produced by genetically modified organisms. Think about all antibiotics that are used in treating infections. Most of them, if not all these days, are produced by genetically modified organisms, right? Now, I may surprise you on this one. Think about diabetes and think about the use of insulin for the treatment of diabetes. Believe it or not, human insulin is produced by genetically modified bacteria. Okay? And by genetically modifying this bacteria, we can produce human insulin these days. Okay? Now, I'm not going to bug you with the very specific technique of CRISPR-Cas9. CRISPR-Cas9, it's actually a technique that allows us today to do genome editing. There is application of CRISPR-Cas9 in, for instance, in solving the problem around sickle cell disease, right? Where one single mutation can be modified to restore the gene. The same thing applies to cystic fibrosis, right? All right, so these days we do what we call exome sequencing. Exome, again, it's the collection of all the segments of DNA that encode a specific protein. Now, exome sequencing is going to give us a good idea as to all the segments that make up for the proteins that are in our system. However, what exome sequencing is not going to be able to do is to allow us to tell specifically what sequence of DNA this genes, the product of the genes came from. Let me close that parenthesis because that can be a little messy there. Okay? When we talk about gene expression, I'm going to explain some more. Okay, so exome sequencing is very valuable. It's very valuable to actually design drugs to treat extremely rare diseases. Okay, now for a long time, it took a long time to try to even read a genome. Now it takes a couple of hours, right, to solve some problems that took days, if not years. In terms of global perspective on genomes, we have to talk about metagenomics. In metagenomics, we are going to sequence the entire genomes that we are going to find in a given geographical area, in a habitat, right? Now, this metagenomics is very important because it allows us to see how organisms are interacting with each other, even to learn that actually some segments of the gene that we find in a human, they actually came from some microorganisms. But that's another story that I'm not going to dive into. Then let's talk about genetic prospecting. Genetic prospecting is the idea that we are going to use metagenomics to see the dynamics of genes in a given geographical area, right? Now, the problem with genetic prospecting is that what is acceptable in the United States, it's not acceptable in Australia, not acceptable in Italy, right? So that you're going to have discoveries that are going to be that are going to be owned by some countries, right? Because, ethics, yes, we talk about ethics, but do we all agree about what is ethical and what is not ethical, right? That's another story, right? So that, some regions of the world are going to literally own some information, and they're going to say, well, US, if you want to use this information, you will have to pay us. Okay, well, that poses a number of questions, right? Okay, how about equal access to genetic tests and treatment, right? How about the misuse of genetic information, right? How about abuses of genetics that at the end of the day can harm a number of people, right? So genetics and genomics can be applied today and allows us to improve the lives of humankind, but also the information. As we get that information, we have to be cautious of how we're going to use it, right? Now, obviously, we are trying to use that information to our own benefit, which is, for instance, for diagnosis, for treatments, for basically defining what is acceptable and what is not acceptable, right? So bioethics is going to be a field that is going to be more and more important as time goes on. All right. So this is where we are going to stop our conversation today. It was a little long, a chapter, but thank you for your attention. Take care now.